As we're going over these different mutations, different types of mutations, and the ways in which they're repaired, I would suggest that you put together a chart of sorts. And what you might want to do is on the left hand of the chart, put the type of mutation. So for example, we started out talking about DNA polymerase errors. These are typically base pair substitution mutations. So we talked about substitution mutations previously. So these are where one base is incorrectly inserted. Oftentimes it's a non-Watson-Crick base pair. And we talked about two different ways that these polymerase errors are repaired. There's the 3' to 5' exonuclease activity of polymerase. So this is the proofreading activity of polymerase. And we also talked about mismatch repair, which involves, in prokaryotes, the recognition of methylated strands as the parental strands from replication and the non-methylated strands as the new strand, and uses the methylated strand as a template to put in the correct nucleotide in the newly synthesized strand. So the next one that we talked about, for example, might be deamination and pyranation, and then you can make it put that on the left side of the chart, and the right side of the chart, put the repair mechanism. Because there are a number of different mutations we're going to talk about, there are a number of different repair mechanisms we're going to talk about. It's important that you keep them organized. So this question here will kind of indicate the importance of repairing these mutations, these genetic changes. So, for example, let's imagine we have a double-stranded DNA sequence, and of course, it's DNA, it's anti-parallel, and we're going to say that this cytosine here is methylated at in this location here. So this is a 5' methylation. So this is now what we call a methyl cytosine, 5-methyl cytosine, and there are drugs that will in fact cause deamination of these methyl cytosines. So if you remember, think back to a few slides ago, what is the base that 5-methyl cytosine becomes when it is deaminated? Well, the base that it becomes is thymine. So loss of this methyl group from 5-methyl cytosine, or loss of the amino group from the 5-methyl cytosine, this deamination results in this becoming a thymine. So if this occurs, what might happen if base excision repair doesn't fix it? So you can think about, after 3 rounds of replication, how many of the daughter cells are going to have a mutation? So take a second, make a prediction, and then we'll go through this. So this is our formerly this is the 5-methyl cytosine. It's been deaminated, so now it's a thymine. And normally base excision repair is going to remove this thymine because it sees that it's paired with a guanine and put in a cytosine. And then after 1 round of replication, both of these are correct. So this base excision repair, this repair pathway utilizes enzymes called glycosylases. And these glycosylases will break, up here, will break the phosphodiester bond and remove this thymine by breaking the bond between the 2 thymines and the bond between thymine and guanine here. And it will remove the thymine and insert a cytosine based upon the fact that the complementary base is a guanine. So after first round of replication, both of these are now going to be correct again, and all the subsequent replication events will result in correct double-stranded DNA sequences. If base excision repair is not working correctly, if there's a mutation in a gene that codes for one of those glycosylase enzymes, because remember, these glycosylases are enzymes that carry out this activity. So if there's a mutation in a gene that inactivates the glycosylase enzyme, what's going to happen is before this thymine can be removed, replication is going to occur. And you'll see that so remember, during replication, both strands act as a template. So the top strand, when this acts as a template, an adenine is going to be inserted in the resulting is going to be placed in the complementary resulting daughter strand. So now this cell has a mutation, and now the cell doesn't have the ability to recognize this as being incorrect because now it's correctly base paired, just like we were talking about previously. So you can kind of look at it after 1 round of replication. After 2 rounds of replication, you can see that all these daughter cells are now going to have the mutations. And these mutations are what we call fixed. So once these mutations are not repaired and replication occurs, they're fixed. They cannot be recognized as incorrect by the cell anymore, and they are permanent. So after 3 rounds of replication, you're going to see half the daughter cells will have a mutation. So this illustrates the importance of these DNA repair pathways, because this is especially a problem in cells that are constantly dividing. So if these mutations occur in cells that are dividing all the time, you're going to run the risk of getting many cells that have mutations. So if in fact base excision repair is not working, do you think you'd see an increase in adenine-thymine base pairs or an increase in cytosine-guanine base pairs after many rounds of replication? Think about this. Think about what the effect would be on the GC content of the genome versus the AT content, and think about how this would work, how you might be able to look at these differences in terms of mutational hotspots. So we mentioned earlier that there are mutational hotspots in the genome where 5-methylcytosine tends to get deaminated into thymine. So would you expect to see an increase in the CG base pair, the ratio of CG base pairs or AT base pairs in those locations of the mutational hotspots in the genome? Think about this. You may see this again.